Hi, everybody. This is Dr. Ed Park from Recharge Biomedical, and today we're going to be discussing autoimmunity, which is, of course, when the body attacks itself. The goals will be to explain autoimmunity, and to do that, we have to explain the immune system. We're going to present my mom a case of rheumatoid arthritis and speculate how this could be helping, if at all. Above me is a diagram adapted from my first book, explaining that short telomeres cause a stem cell mutation and therefore depletion and manifests as many different diseases. And of course, when we talk about autoimmunity, every single type of organ can be affected. So, again, just like cancer, you can get cancer of every organ, you can get autoimmunity of every organ. And indeed, if you live long enough, thousands of years, you're going to. If you want to understand more of my theories of aging and illness, pick up these two books that I wrote. All right, so let's talk about the epidemiology briefly. There's greater than 80 different diseases. It's arguable whether they're actual diseases. They're really just different cell types affecting different organs. Examples above include multiple sclerosis, thyroid problems like Hashimoto's and Graves, rheumatoid arthritis like my mom, which we'll talk about, etc., etc. Another one common is that the GI tract is involved with inflammatory bowel disease. It's said that about 3% of the population is affected by clinically significant autoimmune diseases. And the common ones we know about are type 1 diabetes. That's when the islet cells of the pancreas are destroyed. You can't make insulin. Rheumatoid arthritis, lupus, multiple sclerosis, Hashimoto's, and Graves' disease. Interesting, Hashimoto's attacks the organ. Graves actually produces an antibody that mimics the thyroid stimulation. And anyway, there's so many types of autoimmune diseases. The quote above, "Abandon all hope, ye who enter here," was at the entrance to hell in Dante's Inferno. And I think it's a pretty apt way of saying what Lao Tzu was saying. The more you understand, or the more you know, the less you understand. And that's really true of immunology. I remember I was at, a couple of years ago, at a lecture by smartest guy allegedly in the world, Neil Turok, I think, from the Perimeter Institute. And he was put up one equation explaining all the physics of the universe, and then he said, "Oh, but this is biology, it's much more complicated." And indeed, we see the color code diagram showing just the innate immune system, not even the adaptive immune system, which is kind of an arbitrary distinction because they work hand in hand. But if you just appreciate here, you get pus in the blue, dark blue. You get histamine in the sort of reddish pink. Above, in the yellow, orange, you get cytokines. These things are always active, ready to go. It's the riot police. It's You also have the lipid pathway, cyclooxygenase, lipoxygenase. These things are affected by aspirin and Advil. We have the natural killer cells, which are able to kill stuff without even knowing that it's supposed to kill it. So how does this all work? We'll give you a very brief overview. The human scale distinction is adaptive versus innate. So adaptive, we like to think of as, well, you learn how to attack a new thing, like let's say the coronavirus spike protein or whatever. And so this is a little bit trickier. What we see above me is that you have a naive T cell, which can become Th1 or Th2. Th1 is pro-inflammatory, ramping stuff up. Th2 is a little bit down-regulatory. And we like to kid ourselves that we know how these things react in terms of molecules, signaling proteins called interleukins, etc. But the general gist of it, which they teach in medical school, which is pretty true-ish, is shown to my left, which is that the macrophage will eat up some kind of pathogen and it'll process it into tiny little fragments, and then it'll present that fragment, maybe only 8 or 9 amino acids, onto the surface, where it'll teach a T cell how to kill it if you see that fragment, and then it'll produce B lymphocytes, which will stay in memory. So if you ever see that tetanus or that coronavirus spike protein, you're going to be able to clonally expand, which means, I remember the enemy, we're going to go ahead and expand and overwhelm it. So that's the adaptive immune system. It's both opsonization, which means coating something with these furry antibodies for later destruction, and it's also mediated by the killer cell in the upper middle, which is the CD8. It's trained to kill a certain antigen if present. So the purpose of the immune system is threefold, I guess you could say. It's for critters, cancer, and chaos. And what do I mean by that? Well, just to my left, you'll see a chart of the kind of critters that we can be involved with. These are things you know about. A virus can be intracellular. Okay, so that's the flu virus gets in us, the rhinovirus, the coronavirus, the mumps, measles, they get in us. And so the way to destroy this is not with complement, but you have the natural killers that have to learn to either recognize the interferon signal or to recognize specifically CD8 epitopes. This is fancy jargon, but basically you got to kill a cell that's infected with a virus. Some of the tough ones to get rid of are Listeria and Legionella, rickets, mycobacteria like tuberculosis. They hide inside of us. This also is a function of the innate immune system. Bacteria outside, staph, strep, Neisseria, which is gonorrhea, salmonella. These can be evolutionarily recognized by the lipopolysaccharides on the surface. And so your body has an innate ability to kind of kill those. There's protozoans like Plasmodium, which is malaria, and Leishmania, which is sleeping sickness. And it seems like those have no natural way to get killed, and that's why they're so hard to get rid of. Also, protozoas like amoeba, you go camping, you drink water, you get diarrhea. Giardia, also same thing. And fungi are ubiquitous, and we have ways to get rid of them with the complement and the natural killers. So again, the arbitrary human distinction is there's adaptive, meaning that's the classic thing we see, tetanus, and then you train your B cells, you get a booster shot, you got immunity, and there's memory cells, yada, yada, yada. And then there's the innate, which can recognize unrecognized, future space viruses. So it's quite a clever system that we have to protect us. But it's also protecting us from cancer. In other words, one of the main rules that we don't think about is that when a cancer forms, different behaviors present themselves, which we'll go into a little bit, and that will allow the cancer to die. So the main immune surveillance for cancer is all this amazing adaptive immunity and natural immunity mainly. There's a lot of people experimenting with natural killer cells. You take your natural killer cells, you clonally expand them to millions, take them back, and they just know how to kill cancer. And then finally, chaos is a theme that we go back to in terms of my theory of aging. I think there's three things. There's maintenance, there's creation, and there's destruction. And of course, with the process of living, shutting down genes, getting mutations, we have increasing chaos. In other words, I'm always reminded of these identical twins in their 80s now who were born pretty similar, and then through time, they became different phenotypically, biologically, biochemically, and so that's part of life. You become a stranger unto yourself, and therein lies the problem of autoimmunity. As I revisit this year after year, decade after decade, the things that they taught us in high school and college, they kind of are true-ish, but then they always get revised by a new system, albeit very slowly. So what we see here is the family tree of white blood cells, okay? We got a stem cell, which becomes, of course, the red blood cells, no nucleus. That's pretty clear. Then we got the myeloblasts, which form these granulocytes. Now, the problem is that they kind of categorize things based on morphology, how they look under the microscope, because that was the tech they had, and also function. So in our minds as medical students, doctors, we think of the basophil, the eosinophil, and the neutrophil. They're all what's called granulocytes. And then we have the agranulocytes, which are the lymphocytes and the monocytes. Okay, more than you need to know. And then we have the platelets coming off this other family tree. But the only reason I bring this up is about 12 years ago they changed them all. They said, oh, by the way, those things that look the same, the dendritic cells, which are antigen presenting cells, they can come from either line. They can come from the lymphoid line or the myeloid line. And what I meant to say by clonality is when you really look at the genetic family history, the fingerprinting, the 23andMe or whatever, you see that it's not that simple. They're always revising. And so if you really look at where it genetically came from, the story is much more complex. But let's go back to the simple way of looking at it. B cells versus T cells. So B is for bone, T is for thymus. Okay, simple enough. So we see on the left the bone marrow cells. You have a common lymphocyte precursor. If it's going to become a B cell, which kind of reside in the lymph nodes everywhere, that's going to be the antibody producing cells. Then it'll branch off. If it's going to become a T cell, it goes to the thymus. And what's shown here is that in the thymus, it goes to school. It learns how to recognize self, and these are the biochemical steps involved. They involve microRNA, yada yada. But basically, it has to learn how to recognize self, and it has to learn how to not be overly recognizing and malicious towards self. So again, above we see a classic chart of what we used to be taught is the simplified version. Take-home lesson is you have these things called CD4, CD8, and that's like their cell differentiation markers. If it's a 4 cell, we used to call it a T helper cell. CD8 is a T killer cell, a cytotoxic cell, not to be confused with a natural killer cell. So these things are trained to recognize threats and kill them. So when you overwhelm a cancer or virus infection or what have you, CD8s are targeted for cells that are showing that, as we'll see in a moment. CD4 are helpers. They create memory, regulation, etc. And so, but from both sides of the tree, we have antigen presenting. So what does that really mean? It's very confusing. But let's just revisit the thymus. So the thymus in the top middle is this little, it looks like thyme, I guess, is why the Greeks call it the thymus. It's a relatively big organ in infancy, and then as you get older, it shrinks. So this is kind of where the T cell or the adaptive cell immunity resides. So the bone marrow cells come out and they go to the thymic university or boot camp and they learn. What they learn in the surface or the cortex is how to recognize the self. They need to be able to recognize the self. If they're overly recognizing, they get killed. If they have no recognition, they get killed. So it's like training to be a soldier. But if it's Goldilocks and they have just the right amount, then they'll go to the finishing school, which is the medulla. And in the medulla, again, same thing. They're already be able to recognize the self, okay? But if they're overly reactive, then they're killed. So like in the movie Full Metal Jacket, above me, Paris Island, this guy was tortured and he became a little psychotic and he started to kill his drill sergeant. This is the kind of thing that the thymus takes care of and gets rid of. I think this is the most sciencey that we'll get, and it's a little bit hard to grasp, but just remember there's two kinds of self-identifiers in cells. Okay, there's MHC1 and MHC2, and this is the reason why you have to match these molecules pretty well if you're going to get an organ donation. The more mismatch you have, the more aggressive your body will destroy the organ. So a good way to remember this is MHC1 has only one stem coming out of the cell membrane, and it's got this, alpha, this is sort of 1, 2, 3 thing, and then MHC2 has two. All right, these are both antigen presenting things, which means you don't really present a protein. Like I said, the proteins are diced up and they're presented as these sort of things to recognize. So it's 8 or 9 amino acids. That's why there's cross-reactivity, and so basically, if you look at the chart to my left, you see MHC1 usually takes proteins from inside the cell and presents them. If there are cells that the Paris Island Marine has learned to recognize as normal, it's all good. If it sees MHC1, it tends to think, oh, this is one of our friendly cells. We're not going to kill it. We're not going to frag it. MHC2, on the other hand, is specialized. All nucleated cells, all your cells from any transplanted organ are going to have MHC1, so they better match to a good extent. If you have MHC2, that's only on the antigen-presenting cells like the adaptive immune system. And what that does is it munches up the foreign threat, chops them up into little fragments, and presents them, and they teach each other how to recognize the future threat. So MHC1 is kill me. If you see the signal, kill me, because it's not part of our friendly code. That's how cells will declare themselves infected by a virus or cancerous or what have you. MHC2 is teaching each other, hey, if you see this part of the tetanus or the coronavirus, then kill this. So above me is the final kind of gestalt of cartoons and figures. What we see is that the T cell receptor, that double orange, double blue, 4+4, that's really kind of the handshake with the MHC1 or the MHC2. And that's how the cells present to each other. Hey, I got a virus here, you better kill me. Or hey, if you see this thing in the future on MHC2, you better kill it. Super confusing, I know. The pictures came from this great website, mainbiotechnology.com, and you can see the link there. The further paragraph I thought was really well stated prose, and it says TCRs, or T cell receptors that recognize self antigens, are removed. That's part of that whole training Paris Island. Cell proteins presented by MHC1 should not be recognized by the T cells. However, proteins from a virus or bacteria inside the cell will be processed and presented similarly to normal cell proteins. Since these peptides were not tolerized during the development and therefore TCRs will not be able to recognize these peptides as foreign and identify cells that are invaded by potentially harmful pathogens. So, like I said, in layman's terms, if you get a fragment in the MHC1 and the T cell receptor sees it, but hey, this is not something we learned to not shoot at Paris Island, it's going to take it out. So my little summary here is autoimmunity is often the improper recognition by the T cell receptor of cells who are presenting self-antigens. It either forgot, or it's a new kind of self-antigen, or maybe there's some cofactor from some viral infection. Is it related to viruses, mutation, chronic inflammation? Probably yes, yes, and yes. Again, just to revisit for the final time, MHC1, which all cells have, and all cells can call in the troops to kill them. They can call in an airstrike, so to speak. There's two behaviors. One is it can present a weird thing in the MHC1 and say, hey, kill me, I'm a hostage. Or it can actually not express MHC1, which is a behavior sometimes seen in cancer. So in order to avoid immune detection, the cancer will sometimes not express MHC1. It's like a ghost stealth sort of cell. Luckily, the natural killers tend towards killing cells that don't express MHC1. So it's kind of a hide and seek kind of evolutionary thing. Above me, we see, again, normal cell, copacetic MHC1 with a self protein in it. All good. The natural killer leaves it alone. But if you have a cancer cell, you may have, as shown, an absent MHC1. It's trying to be stealthy, shoot first, ask questions later. And the natural killer goes on. And again, not shown here is if the MHC1 shows some weird, hey, I got some virus inside me kind of signal on the MHC1. That's also tagged for destruction. Okay, sorry about all the science, but this is probably a bridge too far here. But wait, there's more. Everything's so much more complicated. So again, you have the MHC2, which can also be expressed on a tumor cell. So if the tumor cell says, hey, I got cancer here, I got a virus, it can also be killed by virtue of MHC2. So the T cell receptor comes in, and there's training from the CD4, and then there's killing from the CD8. Suffice to say that all you need to take home is that the T cell receptors shake hands with the MHC1 and 2. Antigens are presented in 8 or 9 amino acid epitopes, and it's much more complicated than anyone will ever understand. But basically, you learn and you are able to destroy a great number of different threats, both foreign and domestic. But wait, there's more. We used to think about T cells, the thymic cells. It turns out that the naive T cell, which we test in our anti-aging tests, is CD28. It can differentiate into a bunch of different things. Th1 phenotype, 2, 17-9, Treg, Tr1. This is hopelessly complicated for mere mortals to understand. All these little interleukins and interferons are just approximations. I kind of liken it to this music theory chart above me. You see the well-known circle of fifths, C, a fifth above that is G, go around 12 steps back to F, and you're back at C. Some egghead, lover of music theorist, plotted out what the general harmonies, chord progressions, and structure of the music looks like. And it looks like this for Baroque, classical looks like that, romantic, impression, modern, serialism, I don't even know what that is. Schoenberg, I guess. Okay. Anyway, we're kind of pushing the edge, as Neil Turok said. Biology, especially immunology, especially the ability to recognize self from threat, is super complicated. It's on the order of stuff that we will never understand. And so it's kind of like a music theory chart. So what's the goal? Unfortunately, we're in the medieval times of medicine now. It's sort of like this protester saying, no justice, no peace, abolish the police. Well, we know what happens when you abolish the police. You have no defense. So basically, that's what we're aiming for in a lot of different measured and focused ways, sometimes not so measured, some not so focused. Anti-inflammatory therapies can be somewhat helpful. As I said, those pathways, lipo-oxygenase, cyclooxygenase. Corticosteroids generally inhibit, suppress immunity uh, through T cell function and also through telomerase deactivation. Antibodies can be used in specific targets, although that's a very difficult thing to fine tune. And then there's DMARDs, which is disease modifying anti rheumatic drugs. Basically, things like hydroxychloroquine, sulfasalazine, cyclosporin, methotrexate. These will do specific hits to rapidly dividing immune cells. It's a very crude way to go. If you ever get a liver transplant, it's a double-edged sword because you got foreign tissue in there, a lack of major histocompatibility matching. So you're actually having to take a lifetime of suppression so you don't reject that foreign organ. Okay, let's turn to my mom, Young Park, 83-year-old with rheumatoid arthritis. And of course, exosomes are not FDA approved to treat rheumatoid arthritis or any autoimmune disease. She started to like how she was feeling over the course of the last 2 years. She's gotten 13 different injections. And indeed, we have done a wrist block on her and injected each one of those knuckles, which despite the wrist nerve block was a little painful. I'm not going to lie. But she feels like the movement, the smooth inflammation, the shininess, the swelling, the pain, and even the deformation has improved as a result of just that injection once. We did some of the knuckles that were bothering her a second time, and we also did her knees. So she's a big believer that this helped, at least with the phenotype of pain and inflammation arthritis. Did it undermine or change the rheumatoid factor or the actual physiology of the immune reaction? We don't know. Who knows? So just to wrap up, for those that don't know, exosomes are not cells. They don't have the machinery to make DNA or energy or even other proteins. They're just little messenger packets. They're 1/300th the diameter of the actual cell, and they're extruded in these little packets. And there are ways that the cells communicate with each other. In this instance, it's mesenchymal stem cells that produce exosomes that signal other cells to de-differentiate, to heal, to change their behavior. They don't have self-antigens. They do not have the major histocompatibility 1 and 2. Therefore, they cannot be recognized as foreign, cannot be destroyed, unlike stem cell transplants, which is a common pitfall with that therapy. So finally, how would this work? Well, we see that there's a pretty predictable shift in the macrophage behavior from M1 to M2, M1 being pro-inflammatory, M2 being regenerative, anti-inflammatory. Th1 to Th2 phenotype in those T cells, also the down regulation of the inflammation. We could be regenerating naive progenitor cells. We could be causing death or apoptosis of dysfunctional cells somewhere throughout the body. And we know that autoimmunity is also kind of a function of stress. When you're stressed out, your spleen is activated, you're on high alert, and not only inflamed in your nervous system, sympathetic flight or fight, but this also tends towards revving up the inflammation throughout the body. So they found that if they just stimulated the vagus nerve, it would downregulate the activated macrophages in the spleen. It would downregulate the whole immune system. So indeed, laughter is the best medicine. Relaxation, the craniosacral parasympathetic response shuts all its inflammation down and obviously is good for autoimmune conditions. If you have any questions, you can email me at drpark@rechargebiomedical.com. You can learn more at my website. I just want to close with this thing above, which says cancers and HIV disease. This is a reminder that in the initial days, when I was before I was a med student, they didn't even know what AIDS was, but they found that people would have these rare cancers, and that was a defining characteristic of having AIDS. So, ironically, we know, and we should understand now that we've had this brief whirlwind course, that it's critters, cancer, and chaos. So your immune system is doing an amazing job keeping you alive, not dissolving itself. Yeah, here and there it makes mistakes, and they can be mitigated, but eventually you're going to turn into a chimera of yourself. Your body's not going to be able to remember itself. It's not going to have the ability to reset. So we're heading towards autoimmunity everywhere. But we know that if you don't have an immune system, in other words, if your T cells are infected and destroyed and depleted, this is what you get. You get all these cancers, and they're hugely related to viruses. So it's never that simple. Whenever we were learning as residents and students, they say, well, if you don't know what the heck, you just call it idiopathic, autoimmune, or viral. And indeed, the truth, there may be some truth in that, that all these things that kind of grind down our immune system, especially human herpes viruses, which are indwelling and they live with us forever, um, they're just grinding down your immune system. The CMV, the Epstein-Barr, the herpes. And so we have a depletion of our T cell system, and we have the inability to recognize dysfunctional, cancerous, and harmful cells. And we have kind of like an overwhelming of our immune system. So be careful what you wish for. Immune suppression is not necessarily a good thing. Abolishing the police, not necessarily a good thing. And so what we end up is that we're always in constant levels of dysfunction, of attacking ourselves and tolerating ourselves. And yeah, there are glitches that can happen, and that's just part of the cost of doing business. The reason I put up this picture of Richard is that he's a 48-year-old man, and he looks totally different 2 weeks after exosome therapy. And although his condition had to do, I believe, with leaky gut, we see that because his gut was leaky, he was leaking antigens into his bloodstream. The bloodstream was creating this histamine response. There was swelling everywhere, and he was very sick, and he was having a lot of gastrointestinal problems, varicose veins. So there's many on-ramps to dysfunction. I was talking extensively with a young lady who had Bartonella. And so Bartonella is one of these indwelling sort of bacteria, like some people get Lyme or Listeria, and it's really a drag. And your body's trying to figure it out. It doesn't have the secret formula. It's doing the best it can. But my point is that exosomes, because aging, dysfunction, decrepitude are kind of all going together with epigenetic silencing, gene mutation, stem cell depletion, and dysfunction, senescence, inflammation. Because the exosome phenotype tends to be against all that, it tends to be anti-inflammatory, regenerative, de-differentiation, we tend to have better results. Is this going to mitigate the long-term effect of the autoimmune diseases? Who knows? But when you say autoimmune disease, it's a real garbage wastebasket category. Everything can be recognized as foreign and attacked. So it's kind of it's a misconception to think it's one thing, and so that's kind of what I want to say. This is probably the most nerdy, sciencey, hard to grasp thing, but luckily you don't need to know how any of it works. Your body's doing it for you on autopilot. So with that, I want to turn it over to questions if there are any. I know it was kind of overwhelming and sciencey, and I apologize for that. But let me close the presentation. And if you have a question, you can raise your hand or type it in. I can unmute you. Harry has a question. He asks, exosome treatment for Bartonella, long-term treatment, many sessions. Yeah, I mean, I spoke with one of the YouTubers, Bartonella Babe, and she had some mixed results. Maybe not the most encouraging. Other people are reporting other things. I think for the intracellular pathogens, exosomes have not been a magic bullet at all. I did have the privilege of treating one patient, and I had really very little improvement, and that's unfortunate. But because the community is so desperate, because the immune system is so overwhelmed, overtaxed, exhausted, really, I'm not sure there is a lot of great stuff for that condition. Perhaps they should take a different tack and go massive transfusion of natural killer cells. But that, other than massive amounts of antibiotics in different waves, it's very hard to get rid of that condition, unfortunately. Hello, Harry.
I was just asking for a friend. She's had two surgeries on her hip and scheduled for a third one because she's in great pain and walking with a cane. And I think there's some.
I'm not really sure what two surgeries on the hip means. Do they do like debridement
fell-
of bone spurs, or I mean, we can't really replace the hip twice, so it wasn't that.
No, she fell and broke her hip and had surgery on it and still had ongoing chronic pain after that. And then they went in a second time to clean something up or whatever, and now it's just deteriorating to the point where she's on scheduled for hip replacement and hoping that she can find some way to not have to go through with that.
I mean, without knowing the specifics of her case, my general impression is that she probably had a neck fracture of the hip, and sometimes that compromises the blood supply, causing avascular necrosis of the head. There's a circumflex artery, I think it is, that if you damage that, which often occurs with hip fracture, then the head of the femur dies. So in that instance, there is a transplant they can do. I think it's a fibular graft, but depending on the age and the comorbidity, she may be headed towards a hip replacement would probably be the best thing. Might be unavoidable in that instance.
Yeah, thank you very much. Appreciate that.
You're welcome. Any other questions? Anne, Carolyn? Okay. So, in summary, autoimmunity is ubiquitous, pervasive, accretive, inevitable. So just be grateful that you have an immune system that is keeping you mainly cancer-free, mainly infectious disease-free. I mean, what happens, as we see with AIDS, when you have no immune system, you're just overwhelmed by the natural stuff in you. You get thrush in your throat from the normal Candida that's in your mouth. You get cervical cancer from the normal HPV that a healthy person can fight off. You get Kaposi sarcoma from the human virus. So you need an immune system. You just need one that's self-regulating, and luckily, the thing regulates itself quite well on its own without our help. Okay, everybody, thank you very much for joining. And this has been a webinar on autoimmunity. If you have any questions, you can email me at drpark@rechargebiomedical.com. And I look forward to seeing you on the next webinar. If you have missed any of this, it will be rebroadcast to YouTube after editing. Thank you so much. Everyone have a great day.